to the online webinar which is presented by the RAmediKa and BioKatalyzer. The webinar is on the breakthrough without sacrifice, exploring the future beyond animal testing. Here we are actually going to talk about the innovative practices which are actually like made to remove the outdated practices, that is animal testing. Okay, because we believe that innovation doesn't need a sacrifice. We are going to talk about many alternatives such as medical devices, organ on chips, pluripotent stem cells, and over in silico modeling. Okay, so I will like to invite our speaker from BioKatalyzer, Mr. Chandrajit Dhara. I would like to introduce Mr. Dhara. So basically, Mr. Dhara is a final year BTech biotechnology scholar at the APJ University, Gurugram, Haryana, India. He is currently serving as a member of American Association for Cancer Research and American Society for Clinical Pharmacology and Therapeutics, and also he is an esteemed member of British Society for Antimicrobial Chemotherapy. Mr. Dhara has expertise in cancer genetics, nanotechnology, and medical immunology. He volunteered as a peer reviewer in the Curious Journal of Medical Sciences at the Springer Natures Group in oncology, internal medicine, and medical immunology. Mr. Dhara has published several research papers in international reputed journals, which is indexed in Scopus and Sciences, also in PubMed. One of the example is Spondius publication, Elsevier, and Bentham Sciences. We all know these journals; they are very reputed journals, and Mr. Dhara has like doing great work in the research field, and he is a co-founder in the BioKatalyzer. Now, I would like to hand over my mic to Mr. Dhara to please continue this session and please present your esteemed point of view upon the alternative of animal testing. Thank you.
Thank you so much, Mr. Harsh, for your great introduction. Uh, First of all, a warm welcome to you all for this amazing and insightful session on breakthrough without sacrifice, exploring the future beyond animal testing. So, before beginning my presentation, I would like to know, have you ever thought that why the world is now moving towards alternatives to animal testing?
I believe
World,
it's because we feel that animal testing is unethical.
yes,
It's they are
animal
also a
testing
living being, I
is,
guess.
yes, to conserve the nature. Ultimately, if all the animals or like the in vivo models or the animals will be tested uh, on for the drug development or biomarker development, then there will be one time where all the animals which are being tested till date will be an endangered species, right? Ultimately, the whole ecosystem will be hampered because ultimately the food chain will be hampered and later the ecosystem. So many ethical considerations arise during animal testing. That is why researchers are now moving towards finding the alternatives to animal testing, like uh, induced pluripotent stem cells, organoids, organ on a chips, lab on a chip, and furthermore. So I'll discuss in my presentation, I will talk about the role of induced pluripotent stem cells as an alternative to animal testing. So first of all, we will know about what are induced pluripotent stem cells. They are immature cells which are produced from skin or blood. Then they are reprogrammed into an embryonic like pluripotent state that creates an infinite supply of any human cell which are required as an therapeutic application. For example, induced pluripotent stem cells that can be induced to become beta islet Langerhans cells to cure the patients who are suffering from diabetes. Then blood cells to make fresh blood, fresh blood uh, for the cancer patients who are suffering from leukemia, or neurons to treat neurological problems like Parkinson's disease or Alzheimer's disease. So now the first step to make or design a immunopluripotent stem cell is first is the reprogramming. Reprogramming, scientists are actually reprogramming the cells that have undergone differentiation, like such as skin or blood cells that revert into an embryonic like state. The resulting cells are called, as we know, induced pluripotent stem cells. The patients, the biopsy samples are taken. Then they are cultured as an adult somatic cells. Then reprogramming. Then there are many reprogramming factors, such as some are integrating factors and some are non-integrating factors. Integrating factors, for example, retrovirus or lentivirus. Non-integrating factors such as episomal factors, mRNAs and microRNAs, proteins, exosomes. These are some of the reprogramming factors that are helpful for m- for programming that biopsy or adult somatic cells into the immunopluripotent stem cells or induced pluripotent stem cells, such as hematopoietic stem cells, neural cells, myocytes, adipocytes, beta islet Langerhans, hepatocytes. Then, though these pluripotent stem cells have an application in various avenues of biotechnology drug development, like cell therapy, disease modeling, drug testing, and many more. So now this is a uh, stained images of some of the induced pluripotent stem cells. First is the alkaline phosphatase, the A. Second is the OCT4. The OCT4 is a transcription factor, also known as, it belongs to the POU5F1 protein family that is encoded by POU5F1 gene. It is involved in self-renewal of undifferentiated embryonic stem cells. Third is the SOX2, SOX2. SOX2 is a transcriptional factor that plays a important role in the development of many tissues and organs during embryonic development. Third, fourth is the Nanog Alexa 488. Alexa is the dye. This Alexa 488 and CY3, these are the dyes which are used to stain these cells for the microscopy images. So this is the homobox transcription factor that plays a role in regulating the pluripotency of uh, embryonic stem cells. These are expressed in downregulated. Uh, these are expressed in pluripotent stem cells and downregulated during differentiation. TRA is a gene that in bacteria, in bacteria and in Drosophila melanogaster, and it encodes for the T cell receptor alpha protein in the humans. Now we move to the following uh, applications of induced pluripotent stem cells. As first is its application lies in toxicology testing. Second is in the drug discovery. Third is the drug disease modeling, and fourth is the precision medicine. Ultimately, the precision medicine, it has to work, because ultimately, currently, our world of medicine, medical science, is moving towards the precision medicine, designing precise therapy or personalized therapy for the patients according to their genetic profiles. So next we discuss about what is the uh, what you say, the assay of complete working of an induced pluripotent stem cell. The first process before after the designing or developing of immunopluripotent stem cell for physiology testing or something like drug testing, first of all is the assay development. Assay development there are differentiations or multiple cloning is done, and they are done as three clones per genotype, which are established as a cellular endpoint, which represents a disease relevant phenotype. Phenotype should be robust, it should be reproducible, and it should be amendable. Since screening clones libraries are typically contains the compounds which are dissolved in DMSO, this phenotype, this phenotype should be DMSO insensitive. Then the library selection. The library selection, it should be the chemical screens have opted to use repurposing libraries which contains well sorted, well annotated targets, favorable drug like chemicals to enable rapid transmission into preclinical animal models and human patients. And then the screening of the dose. The screening of the dose, it is possible to perform the screen using multiple clones, and sometimes the typical screens use a single screen at a single dose. This dose can be between 1 micrometer to 10 micrometer. Then the secondary screening is done. In the secondary screening, the dose response, which is measured in EC50, and biological replicates are very important. Then the orthogonal and non-biological reagents are used for alternative reagents or reagent sources to vital to prevent the false positive results. Of course, when we are testing some drugs and all, so false positive results are very much we are prone to get. So the compounds that can be mislabeled or degenerated in library preparation that can mislead the cell or the screening list. Then for that, the orthogonal markers. In orthogonal markers, it could be antibodies, chemicals, or labels that are employed to reduce the compound induced to positive phenotype samples that are irrelevant for the disease, basically. So where there is a case study. There is a QT syndrome modeling where the significant application of iPSCs have been seen. It's a cardiac disorder which can lead to arrhythmia, life-threatening arrhythmia. So researchers have developed cardiomyocytes, which are the heart cells, were used uh, for this study. These drugs, including potassium channel blockers and calcium channel blockers, were tested on these iPSCs, which actually the results, excuse me, yeah. So in this study, the potassium channel blockers and calcium channel blockers were uh, tested on the iPSCs iPSCs derived cardiac tissues. These actually demonstrated that they reliably reproduced the normal cellular phenotypes, which are associated with long QT syndrome that provides insights into drug efficacy and safety without the need of animal models. So this approach not only validates the use of induced pluripotent stem cells for drug screening, but also highlighted their ability to mimic as human disease conditions. Now another case study which is described as chemotherapy-induced cardiotoxicity. It is for the patients with breast cancer and which are the cardiotoxicity symptoms after the treatment of a chemotherapeutic drug, which is doxorubicin. These iPSCs were differentiated into cardiomyocytes to evaluate their responses against this drug. This study found that the patient-specific cardiomyocytes could be accurately predict which patients could suffer from cardiotoxicity due to the administration of the doxorubicin, which is on the chemotherapy-induced cardiotoxicity. It has been tested on the patients with breast cancer, and patients with breast cancer who have been administered with doxorubicin. Doxorubicin is a chemotherapeutic drug. So scientists have developed a immunopluripotent stem cell, uh, uh, which is cardiomyocyte derived induced pluripotent stem cell derived cardiac tissues, which are cardiomyocytes, to evaluate their responses to the drug doxorubicin. And this study found that this patient-specific cardiomyocytes could accurately predict which patients would suffer from cardiotoxicity due to the administration of doxorubicin. This capability allows for the personalized treatment plans where the clinicians have the ability to adjust the doses based on the predicted drug response derived from the patient-specific induced pluripotent stem cells or induced pluripotent stem cell models. Next is the global market, about how big or how giant the global market of pluripotent stem cell production is. So the global induced pluripotent stem cell market would reach around $3.8 billion by 2024. And the market research is also projected that it will reach a volume of $89.5 billion by 2031. And the next big prediction by the global market or market leaders, which are actually developing pluripotent stem cells uh, by about 2 decades, and research is going on, they have predicted that the market will establish from $1.48 billion in 2023 is anticipated to grow a capital annual growth rate to about 9.44% till 2030. So we can imagine how the application of induced pluripotent stem cells have been in the medical industry. We can imagine, right? It is a big market now. It is going to be a big market. So yes, it the market size and the applications and assays, everything can clearly draw attention of researchers, normal uh, human beings, everyone in this uh, planet, that the immunopluripotent stem cells or induced pluripotent stem cell and models are actually the future and could be a possible alternative to the animal testing. As it is easy, as it is convenient, it the researches are going on to make it a cost efficient treatment plan or assay plan that will open the door for precision medicine, as I have discussed. And precision medicine, precision healthcare, personalized treatment is the future in the medical science. So with this, I conclude my presentation and I'm open to the questions you have. Thank you.
Thank you so much, Mr. Dhara, for your kind words. Literally, it was such an insightful that how pluripotent stem cells is going to rule the market in future. Seriously, we all are excited, and I believe that many healthcare professionals, future aspirants, are also with us, and they are also looking this pluripotent stem cells as a very innovative thing to work upon them, and we can definitely see the more alternatives upon the animal testing. And we are open for questions. If anyone has questions regarding this, please feel to ask anything. We are open to all type of questions. And even if your mic is not working, so you can put questions on the chat box. Okay. So Yeah, so first of all, we have a question from Ms. Archita Ranjan. She is asking that how can induced pluripotent stem cell based model be used to study complex disease like neuro degenerative disorders or cancers? Such a nice question. Thank you so much, Archita.
It's really a very good question. So for the neurodegenerative diseases, uh, induced pluripotent stem cells derived neurons. There are neurons which are designed for the treatment of Alzheimer's disease, which are used to study the formation of amyloid plaques in the brain and the neurofibrillary tangles. So neurofibrillary tangles and formation of amyloid plaques are actually a key factors of causing Alzheimer's disease. Second, the modeling of Parkinson's disease. Parkinson's disease, for the Parkinson's disease, induced pluripotent stem cell derived dopaminic neurons that are used to investigate the loss of dopaminergic neurons and the accumulation of alpha synuclein are designed. So, and for the cancers, as in the case study I have actually talked about, that cancer, whenever a patient is suffering from cancer, the main thing that happens is multiple malignancy or allied diseases which caused, like in the case study we have talked about the cardiotoxicity for the patients who are suffering from breast cancer, and they have been administered with doxorubicin chemotherapeutic drug. So induced pluripotent stem cells derived cardiac cells or cardiomyocytes, which have been designed to cure the patients or differentiate the patients. uh, that actually helps the clinicians to identify whether the patient will suffer from cardiotoxicity or not, whether on administering of the doxorubicin. So I hope that answers your question.
Thank you so much, Mr. Dhara. Definitely, the question was very insightful. I hope Archita got her answer. Now the question is from Snigdha. So actually, Srinta is asking like how how can advancement in gene editing technology such as CRISPR-Cas9 system can be used to improve induce pluripotent stem cell based models? That's a nice question about CRISPR and I like pluripotent stem cells. CRISPR is one of the most research topic nowadays. It has been a hot topic for researchers, and it is great to know the insight from Mr. Dhara about CRISPR and like pluripotent stem cells based model. Please, Dhara.
CRISPR, which is the gene editing technique or very advanced gene editing technique, where there are mainly two types of technology by which the CRISPR is happening or which is conducting by the researchers right now. Like there are two, that is HDR, which is homologous recombination repair, and the uh, second is NHEJ, which is non-homologous end joining. So CRISPR is done when there is a defect in the gene, and so we cleave that portion of that gene whereas the defect is there, and the uh, we induce a strand or the correct strand to repair the gene. So it's a gene therapy or a gene repair or DNA repair mechanism. So, induced pluripotent stem cells have been designed or which are incorporated to CRISPR-Cas9, as they are helping to precise the disease models or identify where is the genetic defects or the mutations are happened. And there is generation, we can generate isogenic cell lines. The isogenic cell lines would have the effect on specific gene mutation that can be compared to a genetically identical models. So it would help to identify in which gene or where actually the defect is there or the mutation is there. So the CRISPR-Cas9 technology could be implemented with great accuracy. I hope that answers your question.
Definitely well explained, Mr. Dara.